On behalf of Ashoka, I welcome you all to the public lecture on a half century of cancer research for Professor Harold Varmus, Nobel Laureate, who has been a pioneer not only in cancer research through his own lab, but has also served as Director NIH, Director National Cancer Institute, President Memorial Sloan Kettering, and has thus immensely contributed to advancing cancer research not only in America, but globally. He is currently a professor at Cornell University and is visiting India as the C.V. Raman Professor of the Indian Academy of Sciences, Bengaluru, and I thank the Academy and all the people responsible for organizing his visit. It's a great honor and a pleasure to have the opportunity to introduce you more fully to him. But really, when I look at the CV, the discussions we have had, there is a fear that I might end up taking so much time if I were to introduce him fully that I would be doing the public lecture. And you have come here to listen to him and not me, so I'll keep it very succinct and get out of the way. He was born in 1939 to a medical family. His father was a physician, his mother a psychiatric social worker. We learned a lot about his life yesterday. And in the beginning, he chose a very different track. He went to pursue literature at Amherst College, where he did a bachelor's and a master's in English literature. We are very lucky that his love for words turned out to be perhaps slightly less than his love for discovery and science. And he switched thereafter towards the medical side and went to medical school at Columbia. We also learned very interestingly that perhaps something good, very, very, very silver lining, did come out of the war, the Vietnam War. And as an able-bodied young man physician, he chose to serve the government as a researcher at the NIH, where he worked on cyclic GMP in bacteria to start with. The next phase of his career was working with Professor Michael Bishop at University of California, San Francisco, on a topic that I'm sure we'll hear much about as he speaks. They worked on how viruses cause cancer, and his life could have gone perhaps in either direction. We were talking about that as a virologist or as a person working on cancer. But what he found was that these viruses that cause cancer are using genes that are already present in the human genome. This very important discovery that these oncogenes exist normally inside the genome and mutations by a variety of means can drive cancers was a complete turning point in the world of cancer research. And thus, cancer can occur through the activation of an organism's own genes, and for this, both of them were awarded the Nobel Prize in Physiology and Medicine in the year 1989. Something very, very important perhaps before I get off the stage is that his contributions are not only as an individual researcher or as a group researcher running his lab. They've been on the side of administration where he's advanced the cause of cancer globally. And I'm going to speak more about this globally part later in a panel discussion. He served during President Clinton's administration as director of the NIH, and later under President Obama as director of the National Cancer Institute, and after that as President Memorial Sloan Kettering. In each of these, he has advanced the cause of cancer research across the world. When he took over NCI as a director, there were always many voices about a local view to disease versus a global view to disease, and we will speak more on that. And this use of his stature to benefit other people around the world continues through the public lectures that he continues to give as a distinguished professor. With that, I will get off the stage and invite Professor Varmus to deliver the lecture. Thank you.
Well, thank you very much. Those of you who were not here last night, you've had a good review of what I had to say about my life as a scientist and as a leader of scientific institutions. Today, I'm going to spend almost all of my time focused on science itself as opposed to my own life history. So I appreciate your having heard that background information. Before I begin, I do want to say a word of thanks to Ashoka University for hosting me in a very lively way. Good food, good bed, good company. And I want to thank the Indian Academy of Sciences for inviting me several years ago to be the Raman lecturer. And then because of the appearance of a coronavirus, the trip has been delayed until this year, but that's not the fault of the Academy. So today, what I'd like to do is talk about some of the science I've done in my career, not to just recount these stories and to tell you about what we've learned, but to think a little bit about the route to discovery. How do people discover things that are meaningful? What kind of questions get asked? Why do those questions get asked? What kinds of questions do the answers to the first questions raise? And in the course of doing that, I will proceed from work I did very early in my career, just when at the time I began to think of myself as a scientist, to current day. And I'm going to do this through 6 examples. Hope I have time to cover them all. I think we will. And I want to have everyone here think, even if you don't completely absorb the content of these stories, I want everybody to think about what kinds of strategies actually work for discovering new things about biology in this case, but about life, the natural world in general, in doing scientific work. So I'm going to begin by thinking broadly and historically about how science is done, and then give you an outline of the kinds of problems we're going to talk about, and then tell you something about these 6 examples that illustrate, on the one hand, how in my own life discoveries were made, or in some cases not made, or made by others, and then also tell you, and then leave some time, I hope, for questions before our panel discussion, and then we'll have our panel discussion. So I hope everyone's comfortable, relaxed, and if you need to go to the restroom, judging from last night, there seems to be no peace for the audience, no breaks. So if you need to take a break, 2.5 hours in my own world is too long to sit in one chair. But I hope we'll get through this. So if we think traditionally about how science is done, the beginning of science lies in observation. People in ancient times lying on the ground, looking at the sky and wondering what's up there, looking at plants, looking at animals, categorizing them, looking for strange things buried in the earth. Now, observation is the way science began, but it didn't take all that time before people began to wonder about experiments. What happens if I change the way the world looks? And that led eventually to what we call laboratory experimentation, some of which is driven by hypothesis. If I do one thing, will something else happen? Some of it is just driven by general curiosity. If I examine every blade of grass, how much difference will there be? And some of those ideas may generate new hypotheses, the kinds of things that came out of Darwin's observations of nature, create an hypothesis about evolution, and that creates questions that one might undertake experimentally. And I'm going to be focused mostly in this talk today about hypothesis-driven research, because that's largely what I've done in my own career. But I don't ignore the fact that the kinds of new technologies that have become available for scanning large numbers of people or cell lines or tumors, for changes in genetic constitution or gene expression, have a major role in generating new hypotheses that would then be examined in the way that many of the stories that I will tell you have been conducted. What I'm going to talk about today, though, is how questions that were major factors in my own work were generated. What kinds of things stimulated those questions? What were the factors that led to those questions being vehicles for discovery? For example, was the question just inherently good? Were the methods appropriate for the time and for the question? Were the discoveries important because there was a wide breadth of potential applications in clinical medicine? And were the questions good because the answers raised additional questions that were even better? And as I've mentioned, I have 6 examples. I hope to be able to convey, at least to some of you, the content of each of these problems. Each, of course, is much more complicated than the simple versions that I'll give you in a few minutes each. And hopefully at the end, if there are questions, which undoubtedly there will be, we'll have some time for them. So let me just give you at the outset some notion of the kinds of categories of situation that lead to asking what I think were probably the right questions that were addressed with appropriate tools. So one large category of question gets raised when someone's made a discovery in some other laboratory, and you say, gee, that's an interesting result, but now I wonder about the next stage in the process of understanding some biological phenomenon. I'm going to give you a couple of examples of how that has been a stimulatory factor in my own career. Secondly, there's the question of trying to solve a long-standing mystery. That often happens because a new technology has been developed that allows these questions to be approachable. So there is some mysterious phenomenon. You want to understand more about it. You have some idea of what the question ought to be, and a technology comes along, and you're able to try to answer that question. I'll give you 3 examples of that. And the third category, one that I think is increasingly common these days, as we apply the new tools of genetics and genomics to the study of various kinds of organism, is we find something totally unexpected looking at a large array of individuals or individual situations, and we say to ourselves, how can this be? What kind of mechanism underlies an observation that was made just by surveying large numbers of biological entities? And I'll give you one example from my more recent experimental life that will typify that kind of approach. So number one is here in part for historical reasons. This is not an example of cancer research per se, but it's an example of the kind of research that initiated my own career by showing me what molecular biology can do to help to answer important biological questions. It was my first work as a scientist at the age of 28 when I was at the NIH, as you've heard, trying to serve my country during the Vietnam War without going to war. And I was assigned to a laboratory run by a man named Ira Paston, who's still an active scientist at the age of 91. But Ira had been interested in cyclic AMP, small molecule, which I don't know what the level of understanding of science is here, and I'm not going to have time to describe everything about everything I say, but cyclic AMP is a small molecule that plays an important role in what we call cell signaling in a variety of animals, including human beings. And Ira had discovered that cyclic AMP was not only present in bacteria, but it was able to send a signal to a bacterial cell in a way that changed the expression of an important gene in bacteria. It had been observed by Jacques Monod about almost 80 years ago that in the control of a gene in E. coli, that the gene was repressed when the sugar glucose was present in the growth medium. And Ira had discovered that cyclic AMP, when added to E. coli growing in glucose, induced the expression of this gene called the lac operon. And but it wasn't at all clear what it meant to say that the gene was expressed. As everybody here, I hope, knows, genes are expressed by making an RNA copy of the DNA that encodes genetic information, and the RNA is then translated to make protein. But it wasn't at all clear from Ira's initial experiments whether the level of control was at the level of transcription, when DNA being copied into RNA, or whether the level of translation when RNA is interpreted by ribosomes to make protein. So the question was a simple one. Does what's called the cyclic AMP effect occur at the level of transcription or translation? Or could it be occurring not by the synthesis of RNA or protein, but the stabilization of RNA and protein? And the question was whether we could begin to analyze that problem by measuring in growing E. coli the levels of the rates of synthesis of RNA and protein and the rates of degradation of RNA and protein that are specific for that one gene, the so-called lac operon, or the lac gene. And the problem was that there was no obvious way to measure. You could measure the total RNA in the cell, but you couldn't measure the amount of RNA that was specific for one single gene. And the same might have been true for protein. We didn't get to that stage of the analysis, don't have to, because we solved the problem at the initial stage. And the question is how to do this, make this measurement. And the technical advance was to use a technique that's now known to everybody who's doing modern molecular biology, but it was a novel procedure at that time, and that is to use a technique called molecular hybridization, where you ask, for example, a radioactive form of nucleic acid to form base pairs with RNA or DNA that you want to measure. And by using a couple of tricks that are listed here, but there isn't time to really go into these in detail, it was possible to set up an assay which anyone can see distinguishes one case where the lac operon is being expressed actively, the line that's is rising acutely, or measuring the same RNA in a situation where the lac gene is not induced, shown by the flat line. This is clear that we had an assay that was very sensitive, that could measure even a single molecule per cell of RNA that was specific for the lac operon. And when that assay was used to look at the situation in which the gene is off, the gene is induced by its normal inducer, where the gene is repressed by glucose, shown in the third set, or cyclic AMP is added back in the presence of glucose, you can see that cyclic AMP overcomes glucose repression of the lac operon. And it was doing that, we could show with this kind of assay, by enhancing the synthesis of the messenger RNA for that specific gene. So we knew the answer. Cyclic AMP overcomes glucose repression by increasing the transcription of the lac operon. Well, that seems like a nice resting point, place to stop, write a paper, collect your laurels, and move on. But it also raises some other pretty interesting questions. So how does that happen? How does the RNA polymerase that's making the RNA specific for the lac gene actually get regulated? What is cyclic AMP doing to cells that enhance the transcription of a gene? At that time, actually, there were almost no examples of what we call positive regulation. We knew about regulators that turn genes off, but very little about regulators that turn genes on. So that was one level of question that was actually answered by other people in Ira Paston's lab in due course. But it also raised in my mind, since I was a trainee at that point, what other kinds of problems, perhaps problems more central to my life as a newly trained physician, could be studied with the kinds of methods that I was using. So at the same time that I was working in Ira Paston's lab at the NIH, I was taking courses at night to try to learn more about the scientific understanding of some common important diseases. And one of those diseases I was studying as sort of a fake graduate student, I wasn't really a student, but nevertheless was in class a lot of the time, was that cancer was now being approached with some of these new molecular methods, and in particular, molecular methods that were addressed to understanding viruses that were discovered over the previous 20 to 80 years, viruses that were able to cause cancers in experimental animals, especially mice and chickens and others. And these viruses were interesting just as viral agents that replicated in different cell types, but they were particularly interesting to me because they caused disease in animals that closely resembled human cancers. At this time, 1970, I was asking myself, well, what can the molecular methods that I was helping to develop in studying the lac operon in E. coli that could teach us about viruses that cause cancer. And there were two classes of such viruses, those that contained their genes, their genomes, as RNA, and those that contain their genetic information as DNA. And these two classes of viruses, the DNA tumor viruses and the RNA tumor viruses, all had their advocates and had their advantages and disadvantages. But I became particularly interested in the tumor viruses that carried their genes in an RNA form. And I was interested from 2 points of view. First, how do these viruses multiply? They have their genomes as RNA, that in some sense is atypical for biological systems. So there are a lot of other RNA viruses, including the coronaviruses. And how do these viruses cause cancer? What do they do when they enter a cell that makes a cell behave like a cancer cell instead of a normal cell? And there were a lot of issues that underlie these two fundamental questions. One has to do with the replication cycle. It was obvious that these viruses must attach their genetic information to cells in some stable way because they make cancers that go on to grow for many generations. So that and other kinds of information suggested that their genomes might replicate through a DNA intermediate, which Howard Temin called a provirus, for which at this point there was very, very little supporting information. But it raised the question of whether there was a DNA form of the RNA tumor virus genomes that might be integrated into host chromosomes and hence perpetuated when infected cells became cancer cells. And if that was occurring, there must be a mechanism for making the DNA, integrating it, and then expressing it to make the components of RNA viruses that appeared in the supernatant or in the external extracellular space of these infected cancer cells. These are important questions. I don't have time to begin to answer most of them. I'll focus on a couple, but it's important to point out that although this was a very primitive stage of study of RNA tumor viruses, the fact that many people got engaged with efforts to understand how these viruses work prepared the world in a very important way for the appearance of HIV, the virus that is similar to RNA tumor viruses in virtually all ways and is the cause of the disease known as AIDS. Second big question was how these viruses cause cancer. And there were some really fundamental questions to think about here too. Do these viruses have a specific gene that causes cancer? If so, is that gene required for the virus to grow? Do they use some other mechanism aside from a specific gene to cause a cell to change its behavior and behave like a cancer cell? And these kinds of questions, as you will hear in the next few minutes, laid the foundation for discovering the cellular oncogenes that you heard mentioned in the introductory remarks, and also are now used in making specific diagnoses about what is driving any specific human cancer, and forms the basis for the choice of so-called targeted therapies of human cancer. And I'll mention that briefly as we go along. So this is kind of the background of my second anecdote, which has to do with how RNA tumor viruses grow and how the enzyme required for the RNA genome of these viruses to become a DNA form called a provirus. That was an idea that, as I had mentioned, was first proposed by Howard Temin, an incredibly astute virologist who was working at this time at the University of Wisconsin. And in the early 1960s, Howard had proposed specifically this idea of a provirus, that although no known instance of RNA being converted to DNA had been identified, he proposed that the RNA tumor viruses had some mechanism for converting their RNA genomes into DNA and linking them to chromosomes. And that required an enzyme which had not been seen anywhere in biological systems, an enzyme that copied RNA into DNA and was therefore labeled reverse transcriptase. And Howard and independently David Baltimore at MIT discovered such an enzyme, and in two different kinds of RNA tumor viruses. Those viruses then became known as retroviruses, a term I hope everybody in the audience is familiar with. And they received a Nobel Prize in 1975 for this momentous discovery. But as I was studying these viruses before this discovery had occurred in the Baltimore and Temin labs, I was thinking that I could use the molecular hybridization techniques I had been using to study the regulation of the lac operon in E. coli to look for that enzyme and for protein products that would be made when the enzyme copied the RNA genomes of RNA tumor viruses. But just as I was departing the NIH to start working on these viruses, Temin and Baltimore published their work, and I was left with a good question, but the question seemed to be already answered by these two guys who were some years ahead of me. So what should I do? Well, one thing would be to turn around, go back to NIH, keep working on E. coli, but I realized that there was a tremendous opportunity created by the discovery of this enzyme, namely that finding the enzyme itself was important, but you really wanted to know what the enzyme did. What kind of DNA did it make from the RNA genome of an RNA tumor virus? How was that DNA constructed? What were the sequences at the end of the DNA? How was the DNA inserted into host chromosomes? And there were a lot of really interesting questions to ask as a result of this discovery. And one of the things that was really obvious is that you could use this enzyme to make DNA specific, make radioactive DNA specific for the RNA tumor virus genome, and use that to detect in infected cells the newly synthesized DNA that represented the DNA copy of the RNA tumor virus genome. And that seemed like a really important thing to do. Now, of course, I wasn't innocent enough to think I was the only person thinking about that. A lot of other labs were too. And there was suddenly a new army of people. I was among them who were trying to use the radioactive probes made by reverse transcriptase in the RNA tumor viruses to try to understand how the DNA was made. I can't take you through all those steps and cover the other things I want to talk about, but this picture gives you some idea. This is a complex operation. There was a transfer RNA, tRNA, used as a primer to initiate synthesis by the reverse transcriptase. The enzyme had the ability to jump from the end of a template to another template. It generated two strands. It excavated the RNA template that was remaining to allow a second strand of DNA to be made, and it went through a complicated process that I don't expect you to understand from this complex diagram that led to finding that the product of synthesis was a linear molecule that had some sequences in the RNA genome duplicated at the ends of the synthesized DNA. So that reducing that sentence to one simple diagram generates a very simple view of the RNA tumor virus or retrovirus life cycle, where specialized sequences at the ends of viral RNA are duplicated to form a version of the genome in which there are long terminal repeats, or LTRs, present at the two ends the DNA, which has become integrated into the host chromosome being shown by the wavy lines at the ends. And as you can see, these long terminal repeats at the ends of the proviral DNA have sequences that appear at both the 3' and 5' ends of the RNA. And that can then be used as a template for making more viral RNA that gets used as messenger RNA to make viral proteins and gets used to be packaged as viral genomic RNA that's present in the virus particles of these classes of viruses. Now, importantly, when you look at this simple diagram from the perspective of someone who knows something about movable DNA elements in bacteria, the long terminal repeats resemble elements that are found in mobile DNA elements that can be used as genetic vectors, have the capacity to insert in various places in the host genome, and therefore can serve as we call insertional mutagens, as DNA elements that interrupt genes or activate genes, property of proviral DNA that will become important in a few minutes. But it's significant to note that by studying how viral DNA is made by this exciting enzyme, reverse transcriptase, you come to the realization that something highly specialized and carefully designed is happening, and that there are analogies here that prompt further questions about the biological potential of the genomic elements, the RNA, and especially the DNA of RNA tumor viruses during the tumor virus life cycle. The other important point to take home from this short story is that many of these discoveries were dependent upon discoveries that generate new kinds of technical tools. For example, the discovery of reverse transcriptase itself helped to generate radioactive DNA probes that could be used to analyze the products of the enzyme reverse transcriptase. And that allowed the discovery of many things I haven't had time to talk about. The viral DNA is synthesized in the cytoplasm of infected cells, not in the nucleus. That there is circular DNA, the function of which is still not clearly known in the nucleus of infected cells. That there is integrated DNA in chromosomes. That the kinetics of the synthesis and the copy number of viral DNA molecules can be determined. Later, as restriction mapping became available in the mid-1970s, along with what's called southern blotting, it was possible to describe these so-called long terminal repeats that I've told you about, and that as DNA cloning and DNA sequencing became more sophisticated, it was possible to examine the junctions between viral DNA and host DNA at sites at which integration has occurred. But that's all for some other time. Now, at this point, I want to say a word about reverse transcriptase itself and how it is made during the virus life cycle, because that revealed yet another surprising feature of these viruses, one that, surprisingly enough, proves to be relevant to the current pandemic that we're going through. And let me explain why I say those things. So by the early 1970s, people began to think about the genetic composition of the genomes of RNA tumor viruses or retroviruses. And in the case of the chicken retrovirus that my own group was working on, the Rouse sarcoma virus of chickens, it had been possible to say there are at least 3 major coding domains that are important for the growth of the virus. An element called GAG, which happens to make the proteins that are found in the inside particle of the virus. A region called POL stands for the RNA-dependent DNA polymerase, or reverse transcriptase, and a region called envelope, which makes the glycoprotein that's present on the surface of the virus. And a 4th element, which we'll talk more about in just a moment, called SARC, which makes the protein that is important in generating sarcomas when this virus infects a chicken cell. And if one wanted to understand how this virus makes reverse transcriptase, the protein that it needs for copying its RNA genome into DNA, you would find that function maps genetically to the middle of the viral genome, hence it's called POL for polymerase. But when you look for that protein product using antibodies against reverse transcriptase, you find that there's only a very large protein that seems to be the precursor to the mature reverse transcriptase. There's no shorter protein made. And the only shorter protein is one that's made from the upstream region, which is called GAG in these diagrams. And that raises an important question. Where does the polymerase come from? How does it get synthesized? And it was clear in the late 1970s, when it was possible to determine the sequence of these genes, that there was a gene that could make GAG proteins, and there was a gene segment, an open reading frame that could make polymerase, but they were in different reading frames. And yet there was no, if you looked at the RNA made from the provirus, to look at messenger RNAs, the only RNA that seemed eligible to serve as a messenger for the very large protein that had both GAG and POL elements was RNA that was indistinguishable from full-length RNA that would be found in the virus particle. So that raised an important question. How do you make reverse transcriptase on the ribosomes? And there seem to be only two possibilities. One is that the RNA is made, that the polymerase is made from some atypical RNA that's been spliced to generate RNAs in which GAG and POL are in the same reading frame, or ribosomes themselves have the ability to recognize the need to change reading frames as they're synthesizing proteins. Very hard to, this is a question that's been debated by many students of these viruses for several years. It wasn't at all clear how to resolve this problem of finding a low abundance, atypically spliced RNA that could be the messenger RNA for making the GAG-POL fusion protein, or did the ribosomes change reading frames? What kind of RNA would you use to analyze that? This problem was solved by a very clever experiment done by a guy named Tyler Jacks, who was then a graduate student in my lab, more recently been the head of the Cancer Center at MIT. But Tyler proposed something quite clever, namely, let's just synthesize full-length RNA from cloned retroviral DNA of this virus, the Rouse sarcoma virus, and synthesize it with a bacterial RNA polymerase and a system that couldn't possibly splice the RNA, and then translate that RNA in a mammalian translation system for making protein, and then look to see what kinds of proteins are made. And lo and behold, to I think everyone's surprise, both the GAG protein, the shorter protein, and the longer GAG-POL protein were made in the correct ratio in that in vitro translation system. And that led to the obvious conclusion: the ribosomes must be shifting frame in the -1 direction to make the GAG-POL protein. And we looked at this more closely to try to figure out why ribosomes were shifting frame at one particular point in translation. We found a characteristic sequence with a certain set of nucleotides called the slippery sequence, and downstream in the RNA from that slippery sequence was a place where RNA could fold up into a structure called a pseudoknot, and the pseudoknot induced stalling of ribosomes at that phase, and the slippery sequence shifted the ribosomes back one nucleotide while trying to make protein. And this was not only an interesting finding, it was one that could be extrapolated to other situations, because, as could be predicted from nucleotide sequencing and was validated by actual experiments, other RNA tumor viruses, retroviruses, did more or less the same thing as we saw with the chicken retrovirus, Rouse sarcomavirus. That's true of HIV, the mammary tumor virus, and others. And it turns out that there are other RNA viruses, including the coronaviruses, that direct ribosomes to shift frames. And indeed, even the coronavirus we're all worried about in this room, SARS-CoV-2, has the requisite slippery sequence and pseudoknot downstream that allow the coronaviruses to make some important predictions about viral evolution that are worth thinking about. It also suggests an avenue for developing drugs against these viruses that have not yet been successfully pursued. So, end of story 3. Let's say something about the next example of a mystery that had been long-standing and unresolved when in the mid-70s, Mike Bishop, my colleague that you've heard described, and I began to worry about why retroviruses like the Rouse sarcomavirus of chickens might carry an oncogene. What we knew from evidence I'll show you in just a minute that there was indeed a gene I've mentioned before called SARC that was responsible for initiating the cancerous change that results in the production of sarcomas when chickens are infected by Rouse sarcomavirus. But it wasn't at all clear why the virus would care about having such a gene, because we already knew that even in the absence of the SARC gene of Rouse sarcomavirus, the residual virus shown on the right in the diagram at the bottom was able to grow at very high titers. So why would this gene be, even if it does cause cancers, why would the virus care about having a cancer-causing gene in its genome? And that gave rise to a lot of thinking and talking. There were some clues to what might be going on here, namely, first of all, that we know that unlike the situation with the DNA tumor viruses, retroviruses didn't need genes like SARC to multiply efficiently. Rouse sarcomavirus is a virus that arose only rarely in nature. There have been a few isolates of this kind of avian sarcomavirus, but doesn't happen very often. And each occasion seems to be novel. We knew from studies that I won't describe here is that retroviral genomes can recombine efficiently. So there's something about reverse transcription having to do with the ability of the enzyme to jump from one template to another that might account for a recombination phenomenon that would mediate the capture of a gene from some other virus or some other cell. So this gave rise to a hypothesis. Could the oncogene of Rouse sarcomavirus, the so-called VSARC gene, have been derived from a gene in which a virus lacking that gene was replicating? And could we actually test that idea? In the early 1970s, it became possible because of the existence of reverse transcriptase itself and because of fancy molecular hybridization techniques to actually do an experiment to test the idea. And then we asked ourselves, how would we interpret those results? So let me remind you about a few things concerning Rouse sarcomavirus. As I mentioned, this virus was discovered very infrequently. First discovery was in 1910 when Peyton Rouse at the Rockefeller Institute was given a chicken carrying a cancer called a sarcoma by a local poultry farmer, and he put an extract of this tumor through a filter and identified a very small particle, which became known as a virus, and that particle was able to cause tumors again when injected into a chicken, and then to overcome the difficulties of working with animals many years later. Two investigators, Howard Temin, who I already mentioned, and Harry Rubin, showed that there could be developed a simple test for the tumorigenic abilities of this virus without having to inject chickens and cause sarcomas. And that simple test involved growing chicken cells in a petri dish, infecting the petri dish, the cells in the dish with Rous sarcoma virus, and seeing a focus of abnormal cells that have the ability to cause cancers when put into an animal. That assay for the oncogenic effect of this virus was then the assay for developing mutants that showed that there was a gene in this virus that was essential for its transforming abilities, and that genetic test was most powerfully displayed by work of a postdoctoral fellow named Steve Martin in 1970, who isolated one of the most powerful and informative temperature-sensitive mutants of Rous sarcoma virus that showed that a virus with a mutation in the SARC gene could replicate perfectly well at both high and low temperatures, but it failed to transform cells at the higher temperature. Moreover, if you switch the temperatures up and down, you could show that that when you allowed the gene to function at the lower temperature, the cell was rounded up and behaved like a cancer cell. But when the temperature was raised, even though virus was continually produced, the cell reverted to its normal behavior, suggesting that continued production of the protein product of the viral oncogene, the SARC gene, was required to maintain the cancerous state. But the gene was not required for replication. So that raises the question that we're thinking about here. Why does the virus actually carry that gene? And maybe the gene really isn't, it's not necessary for the virus. Maybe it has the gene only because somebody interested in cancer isolated this virus and decided to work with it because they were trying to understand cancer. And the ability to pursue this question, seeing whether that gene actually was derived from a normal cellular gene, was made possible because there were other kinds of mutants of Rous sarcoma virus that had deletions in the viral genome that encompassed most or all of the viral SARC gene. And by using such mutants shown here in the box around the gene SARC, it was possible to make a radioactive piece of DNA that was specific for that deleted region that contained most or all of the SARC gene. And that was done by a simple method of molecular hybridization that I won't get into in detail. But once that radioactive probe that was specific for the cancer-causing gene was used to look for that gene in normal cellular DNAs, it was possible to show that while the gene was present in chicken DNA and many other types of animal DNA, and other viral genes happen to be the confusing aspect of all this. Is that many of the other genes of the virus are present in chicken DNA because animals like chickens do have what are called endogenous viruses carried in their genomes. But the important point is that the SARC probe detects a gene which is present in essentially all animal species and is highly conserved. And the way we think about this, even if you haven't absorbed what I just told you, is that the transforming gene of Rous sarcoma virus, shown on the right-hand side of the 3' side of the viral genome here, is derived from a normal cellular gene that has introns and exons, and was captured by a virus that was able to incorporate its genetic sequences into the genome through recombination. That means that there was a viral version of the SARC gene that encodes a protein, and a cellular version of the SARC gene, which encodes a very similar protein, but the proteins differ in their enzymatic activity. The genes differ by a few nucleotides that changes are responsible for differences in the enzymatic activity of the products of the viral and the cellular version of this gene. And that accounts for the ability of this virus to cause cancer and the capacity of the normal cellular oncogene to participate in developmental events in the normal behavior of the normal development of a chicken. So what we've learned by asking about these complicated questions about the origins of the transforming potential of the retrovirus known as Rous sarcoma virus is that the virus carries a cellular gene. That gene does happen to encode an interesting enzyme, which we don't have time to discuss, a protein tyrosine kinase. And that gene was derived from a normal cellular gene, stolen, if you will, and it raised the question of whether other cancer-causing RNA tumor viruses might have other genes that are derived from normal cellular genes. And indeed, as this iconic cartoon shows, that is the case. There are many other retroviruses that cause cancer in a variety of animals, mainly mammals like mice, cats, and rats, and even non-human primates, and that those genes are all derived from normal cellular genes that have been captured during the growth of a retrovirus that lacked an oncogene. Now that raises questions about what roles these genes might have in human cancer. And one of the great themes of the last 30 years or so of cancer research has been to show that many of these genes that were discovered by using animal retroviruses that carry viral oncogenes are mutated in human cancers and have been the targets for development of drugs that are now widely used in clinical work. So clinical cancer has been the care of patients who have cancers has been forever changed by the identification of mutant genes like a mutant so-called ABL gene and mutant forms of so-called RAS genes, and the development of drugs that inhibit the activity, the function of those genes to change clinical control of human cancers. And I just remind you, this all derives from asking a fairly simple question: why is there a Sarc gene in Rous sarcoma virus? Why is there a RAS gene in murine sarcoma viruses? And where do those genes come from? And do those genes ever get mutated in human cancers? Now, let me say a few things about a corollary that might have occurred to some of you. I've mentioned that there are a lot of RNA tumor viruses that don't seem to carry an oncogene. And a general question was raised as we began to understand more about genes like Sarc, and that is, how do the RNA tumor viruses that don't have a viral oncogene derived from cellular genes cause cancers? I show on the right side of the slide the Rous sarcoma virus genome that we've been discussing, with its replication genes and its Sarc gene for inducing sarcomas. But there are lots of other retroviruses, including, shown from top to bottom on the left, the avian leukosis virus. That was a way of demonstrating that LTRs are transcriptional enhancers that can be inserted near a proto-oncogene in either direction on either side of the gene and cause activation of that gene. So that was all useful. It was also a way to promote our excitement about going to identify the gene that might be activated by the mouse mammary tumor virus. We carried out an experiment that was, in a sense, unbiased. We didn't worry about what the gene we might be. Instead, we went ahead to try to clone the gene by a procedure known as transposon tagging, thinking about the provirus as a transposon, cloning the mammary tumor virus provirus and looking at the DNA next to the inserted provirus, which had been activated transcriptionally by the insertion. And the result was to show that the provirus of the mammary tumor virus that induced breast cancer in mice was an unrecognizable gene. It had never been seen before in the genomes of any RNA tumor viruses. And Roel Nusse, who did this work as a postdoc in my group, initially called the gene for its integration site, INT1, later turned out to be closely related to a gene in Drosophila known as wingless and was called the Wnt1 gene, a gene that we now know to be capable of making a small signaling protein that's secreted and turns on a signaling pathway that changes the transcription of host genes. Let me just try to summarize what was learned by this sequence of events of trying to understand how an RNA tumor virus without an oncogene can cause cancer. First, we and others learned that the avian leukosis virus that lacks an oncogene can cause a B-cell lymphoma by inserting its provirus next to the c-Myc proto-oncogene, and now many people have shown over the last 30 or more years that the c-Myc gene is very commonly perturbed in the course of human cancer development. And though there are no effective drugs as yet that address cells in which the c-Myc gene is perturbed, nevertheless, we know that c-Myc is an important therapeutic target. In contrast, the Wnt1 gene is another proto-oncogene not found in any retroviruses, and turns out not to be found activated in any human cancers. But are the Wnt genes actually instructive about human cancers? And the answer there is yes, that Wnt signaling pathways contain a number of other proteins that are involved in human cancers. And virtually all human colonic or rectal tumors do have mutations affecting genes such as the APC gene or the beta-catenin gene that play an important role in cell signaling by Wnt genes. Now, I was expecting to give one last story, but I think given the time constraints here, I'm going to just skip to the end here and tell you what I think we've learned about scientific discoveries. My goal here was to communicate some information about human cancer and how we learn certain things about human cancers by studying RNA tumor viruses. But I do want to emphasize here, because only a few of you here are cancer biologists, to make some fundamental points about scientific discoveries. The scientific discoveries begin by identifying an important problem that is right for study at that time. So then it's making progress against the problems that are presented by some biological situation, like conversion of a normal cell to a cancer cell, only get made when you ask the right question. And then that you have the right method available for trying to answer that question. You confer the right interpretation on the message. You have the tools for following up the answers to the first good question by answering additional questions. And then you find appropriate applications, like using targeted therapies to understand, to try to interfere with the function of activated cellular oncogenes in human cancer. So my message here today is that science is a powerful tool that allows you to do some extraordinary things. Ultimately, we'd like to benefit the human condition through the science that we do. And it does take an appreciable constellation of events, discoveries of various kinds about how viruses grow, ways to understand how viruses like the RNA tumor viruses with or without oncogenes cause cancer, and then an effort to understand through a community effort what the implications of discoveries are for the diagnosis and treatment of diseases like cancer. So let me conclude here and take some questions, and then we'll move on to the appointed panel discussion. Thanks very much for your attention.